Okay, I guess I'll just start. I'm Kristina Hillesland, for those of you who don't know me, and I am a professor at the University of Washington, the Bothell campus, which is part of the UW system, but we don't have grad programs, so it's a primarily undergraduate institution. And so you're seeing like the most sciencey looking thing in my lab in this picture, and I'm going to tell you a little bit about how I use big blow up devices like this to study how positive associations between species evolve. So when I was in grad school, I sat in on this symbiosis class at Michigan State in the microbiology department. And we learned about this particular mutualism that is shown here. I apologize for the old fuzzy picture, but what we have in here is a bacteria, which is this green fluorescence, and an archaea, which is the red fluorescence. And this little consortia of these two species, they live like way deep down in the bottom of the ocean. And for a long time, people were wondering how they lived together in these environments where there was no oxygen and very little energy sources. And they figured out, we call these anaerobic methane oxidizing consortia, but they figured out that what happens is that the archaea takes methane and like makes carbon dioxide out of it, which is kind of the opposite of what we normally think of archaea doing. It's like a backwards reaction. And to make that go though and get energy, there's the bacteria reduces sulfate to hydrogen sulfide. And by sort of doing this big chemical reaction together, they're able to survive, and they grow really slowly. And there are a lot of little things they could notice about them, like there was always the same ratio of green to red in these little consortia, and they can actually culture them in the lab, but they've never been able to like separate the bacteria and the archaea from each other. And so I just found this really interesting, because usually when we learn about symbioses, we learn about like an animal with like a microbe symbiont or something like that. And I thought this was interesting to think of two microbes that are working together. And of course, being from an evolutionary biology lab, my interest in it was really about like, how do these things evolve? Not necessarily where did they come from, but like, how do they continue to evolve in these situations? And I was studying predator-prey interactions at the time, and I started to develop this interest in thinking about like, well, what happens when you have two species that are interacting and they're actually just having positive effects on each other's fitness? How does that change? Like, what kinds of adaptations might occur? The rate of evolution, and all kinds of different features of the process and outcomes of evolution. And so this question that I've been trying to address for actually a long time is like, how do species adapt to one another when they have positive effects on each other's fitness? And we know a little bit, right? So there's all these examples of studying endosymbionts where we can see that they lose functions that are no longer needed. Like a key example when I was first learning about this was Buchanera, that has like these like tiny little genomes after millions of years of living inside of their aphid host. And we think this also happens in microbes just in the wild when they are in interdependencies. Another possible thing that we know is that sometimes when there are these positive interactions, one species, there's a new niche that becomes available, and one species might adapt to that. Like, for example, you know, like bobtail squid have these special compartments where they hold fluorescent bacteria. And there are these ants that have made these special little crypts to hold fungi that they use to cultivate their little gardens. So they're adapting to having this species that is helping them out. So we know that this happens. And actually, one thing I've always really been interested in, and I'm going to show you some of my data on this, is how often and in what situations there would be opportunities for coevolution to occur. So of course, I needed to study something that could be cultivated in the lab where the species could be separated, and where I didn't have to go to the bottom of like super deep areas of the ocean and collect them on a regular basis. So I found out that through the grapevine that at University of Washington, Dave Stahl was sort of making a mutualism that is kind of similar to the one I just described. So in this system, we have a bacteria, Desulfovibrio vulgaris, and this bacteria, we put this in this special conditions where it has to live by fermenting lactate. And when it does that, it makes acetate and hydrogen and bicarbonate as byproducts. And this reaction actually barely provides even the tiniest bit of energy if you just have it in standard conditions where all the reactants and products are at equal concentrations. And so you would kind of, so if they're living alone in these conditions where we are growing them in, they won't live for very long and they won't be able to like make more copies of themselves. And so we add this archaea, this methanogen that consumes the hydrogen that is produced by the bacteria. And this does, first of all, it's the only hydrogen available to the methanogen. It's its only energy source. So, so that provides food to the methanogen. But the other thing it does is it draws down the hydrogen concentration in the media. And that basically changes the energetics and pushes the reaction forward and allows the bacteria to keep generating energy. So if this is like a lot of chemistry for you to think about, if you're not used to it, some of us aren't, that's okay. You might just think about it that like the bacteria feeds hydrogen to the methanogen, and if there's too much hydrogen, it sort of becomes poisonous. And so the methanogen, by eating the hydrogen, is sort of like keeping that environment detoxed. So both of these species are lab strains, and they were isolated like in the 60s and then propagated by different labs and studied a lot in those labs for a while for unknown lengths of time in between being frozen down. And they came from really different environments. The Desulfovibrio came from a freshwater and the methanogen from a brackish environment. One of them was isolated in like England, and the other one was isolated over here on the Pacific Ocean. So I don't think it's likely that those particular strains co-evolved for like a long time just before I started using them. And then also, you know, I can change the environmental conditions in the media and propagate each species separately, which allows me to do lots of experiments. And then the last thing I want to say about this system is that I can't really say this for sure, but I think a few theoreticians and other people have thought that perhaps this sort of might be a cheat-proof mutualism. Because there's really only one thing that's being exchanged, that one physical thing, which is hydrogen, which is not costly for the bacteria to produce. And it's in the best interest, we think, of both partners to like maintain hydrogen at a low level. So, I mean, I can't say for sure, and I have some alternative hypotheses, but we haven't seen cheating yet. And, you know, so we can kind of think of this as like a real positive interaction, perhaps. So many years ago, I started this evolution experiment where I put one of each species into a tube and I made a bunch of copies of it, and I let 24 cultures evolve, and some of them were shaking really fast, so the hydrogen is dispersed by just all over the place all the time, and some of them were just incubated upright. So the hydrogen mostly accumulates in the headspace and is highest concentration near the surface of the liquid. And then I've also, over the years, developed some controls for this. So I can evolve the methanogen alone. All the conditions are the same, except we have to add hydrogen, otherwise they won't have an energy source. And because there's acetate in the media that is made by the bacteria in the mutualism environment, we add acetate to our control so that they have that available, which they can use as a carbon source. And then we also have the Desulfovibrio, which is evolving alone. And we use, it can't live by that fermentation reaction by itself. So we provide it with an electron acceptor, which is sodium sulfite. So what we found out in studying the mutualism is that within the first 1,000 generations, they evolved so that they grow a lot faster. So in this graph here, oops. Okay, the ancestors are blue, and you can see this is their growth rate. And then all of these other colors are basically have either species evolved paired with the ancestor or both of them together. So each of these is a co-culture. So for example, co-culture U12, this would evolve, it grows faster with than the ancestor, regardless of whether it's the disulfur vibrio, the methanogen, or both that are have evolved in syntropy. So, and in fact, they grow a lot faster. The ancestor would double about every 20 hours when we started this experiment, and now at 1,000 generations, they doubled about every 5 to 8 hours. And actually, we haven't tested it out as far as 5,000 generations, so I don't know if they're growing faster still or not. And they also produce more cells. Once again, if you just look at the colored stuff relative to the blue, you can see that we have a higher density. Okay, so they've clearly changed. So how? How do they adapt to mutualism? Well, this was still when I was in Dave's lab when we first started this, but we sequenced a lot of these co-cultures at 1,000 generations, sort of like little mini metagenomes. And one of the biggest changes that we saw that was really common had to do with the disulfur vibrio, which I labeled D here. Okay, and this D is a sulfate reducer. It's like, that's how it lives. It's sort of a way we categorize things as microbiologists. It's sort of like saying that fern is a plant or something like that. That's how we calculate it or talk about them ecologically. And so what that means is that they use sulfate biochemically in a way that's very similar to the way that we use oxygen as an electron acceptor. And to do that, they have to reduce sulfate in 3 steps with 3 different enzymes. One is the SAT, one is the APS-AB, which has 2 protein subunits. And then there's a 3rd enzyme that goes from sulfite to H2S. And that one is the dissimilatory sulfate reductase, and it has 3 subunits. So that's some background. And this is, this sulfate respiration is one of the main reasons this disulfur vibrio has been studied for years by many different labs. And so, it's an interesting trait to us. But in the evolution environment, there was no sulfate present. Right? So guess what happened? When we look at all these different little mutualists, we can see these are each of these little boxes here is a different co-culture. And these are just the ones that we sequenced. We also have data showing that this happened in other of the evolved co-cultures, little mutualisms. And these red bars are showing that, like this one is showing that most of the population of culture U9 has a nonsense mutation in the SAT, this first enzyme here. And then there are also lots of co-cultures that have nonsense mutations in either, the APS-A or B. And so, and these are dominant in these populations that have these nonsense mutations are dominant in these cultures. And they've remained dominant for a long time. And so we saw a lot of examples where they no longer can respire sulfate. So another thing that you might notice is all these blue bars, which shows that this C subunit of this final enzyme has a lot of missense mutations, which we think is interesting, but no one has followed up on that, and we really don't know why yet. But if there happens to be a biochemist in the audience, they might be interested. I don't know. The other thing I wanted to mention is, remember I told you I had this control experiment today where I just had the disulfur vibrio evolving alone? Like, one of the questions could be, is it just because there's no sulfate, or could it have something to do with mutualism? Could there be a trade-off here? So in these conditions where the disulfur vibrio is evolving alone, it doesn't have sulfate, it has sulfite. So it has this, and it can only do, has to do this last part of this at this pathway. In those conditions, it has maintained all of these sulfate respiration genes. So in the first 1,000 generations at least. So there's a lot of potential explanations for that, but I think it's interesting. I was kind of glad that it's not just like lack of sulfate, but perhaps something more. Okay, so this tells us that there's some adaptation, that there might be some cost to maintaining sulfate respiration in this environment for the disulfur vibrio. But what I've always kind of really wanted to know is if these species can adapt to each other specifically, or if they're just sort of adapting to sort of this general chemical environment. And so one of the first things I did was try to see if they are specifically adapted to the partner they evolved with. And so this is kind of an easy experiment to think about, which is just to take cultures that evolved together, separate the individuals, the two species, and then mix them back together with partners that evolved separately. So that's essentially what I'm showing here. So you have, we have species pairings that are local, where they evolved in the same tube for 1,000 generations. Or we can take, like, say, a disulfur vibrio from one tube and a methanogen from a different tube and pair those together and see if they grow or have a similar fitness. Or we can even take them from different tubes and also different environments. So like one that evolved in the shaking environment and one of them that evolved without shaking, and we compare those together. And so we made a lot of these pairings to see if when they evolve together, do they have higher fitness than they would if they were paired with these individuals that evolved with other partners. And we've tested this in 3 ways, but in the interest of time and the fact that some of this hasn't been analyzed properly yet, I'm just gonna tell you the results of looking at absolute fitness. So in this case, what we're doing is we're letting these different cultures grow up until they use up all the resources. And then we're looking at the relative abundance of each species. And they look very different from each other under the microscope. So we don't have to do a whole bunch of fancy extra work to be able to just look under the microscope and count each one, which is rather nice. And so it's sort of like saying, testing whether their carrying capacity is a little bit different or their absolute fitness. There's a couple ways to think about it. So the answer is basically that well, I'll just tell you. Okay, so we had like 72 different pairings that we've studied from 12 different evolved communities. We've replicated it 6 times. I've had like 5 different student groups work on this, and in a class that I teach, and over time. So we have a lot of data. And here is an example of it. These are just the means, but I've also like run some mixed model analyses to test whether or not these are different, accounting for all kinds of things like differences between students and times and all kinds of things like that. So this is the population size of disulfur vibrio when it is paired with a distant partner or a local partner that it evolved with, and you can see that the means are like, you could practically just superimpose those 2 boxes right on top of each other. The only difference is that there's a little bit more variance, and this is this idea that there's no difference between them pans out pretty clearly in the model. It's like a p-value of 0.948. So they're the same. There's no differences that we can do. And if we looked at Methanococcus population size, we get the same thing basically. There's more variation when they're with a partner that they didn't evolve with, but there's also more samples because of the nature of the fact that of the experiment. So the one difference that we can see in this experiment is that when disulfur vibrio is paired with a methanogen from the shaking environment, where the hydrogen is being dispersed by shaking, when it's paired with a methanogen like that, it has a slightly higher population size than it does when it's paired with a methanogen from a non-shaking environment. So, but we don't see that for the methanogen. So it seems like there's some kind of adaptation of the disulfide vibrio that has to do with this shaking environment and whether the methanogens were in that environment as well. So, all right, so that data is complicated, but also slightly not very encouraging about the possibility that they're adapting to each other on its face. But I still wanted to know a little bit more, because what could happen is they could be adapting to each other, right? In all of these independently evolved cultures, but maybe they're all adapting kind of in the same way. Like maybe they're all adapting to each other, but they're sort of fixing the same kinds of mutations or having the same phenotypes are changing. So then if you're swapping them, they're more or less similar. So to test that a little bit more detail, we've been looking to see if they've been co-evolving in this first 1,000 generations at all. So they could have been co-evolving together, but along similar paths, different tubes along similar paths. And so maybe, maybe that's happened. So that's what I wanted to test. So I'm going to just kind of tell you a little bit about coevolution and the way I think about it, and then how we tested it. So imagine that you have two species, and one of, oh, I keep pointing with my pen and forgetting that you can't see that. Sorry. You have two species, A and B, they're in a mutualism, and they're trading a resource or two. And species A gets a mutation that makes it A1, and now it's making this like this little miniature like car that can trek the resource from A to B. So now this would be kind of an example of like this new niche idea where B now has a little car that it can adapt to. So maybe it adds this little yellow box, which could either be keeping the resource from A, its resource from A, or helping to hold things in a little bit better. And then A could adapt to that little car now too, and add trick it out with some green stuff. And then, you know, you could have this going back and forth for a while. I mean, I think that this is sort of like my initial view of coevolution, is that you have these, this back and forth of evolution, and where they're continuously adapting to each other's changes. And so at the end, you can have this really complicated Lego car that does a great job of transporting nutrients between them in some way. So that would be coevolution, but you can also have species that they can still adapt to each other without coevolution. You can have co-adaptation. So in this case, A would still get this, you know, little car, and that would help it to adapt to species B. But for whatever reason, B doesn't adapt back. So it just stays, and the car always stays in this sort of blue and white configuration. So either of these things these are all examples of the way that species could adapt to each other. And so to test this, what we do is go on this idea. So imagine that you have BX that makes the little yellow box, and you pull that one out, that intermediate out, and you pair it with A1, which makes the predecessor for the little yellow box, and you put them together, right? It's going to be happy, and you're going to have you have them interacting with each other, and they're going to have this little car with the yellow box because they have all the pieces. But what if instead you take BX and you pair it with like this A1 through infinity over here that has all these pieces to make this car? In that case, all these intermediates to go from here to here are missing. And so you might find out that you can't get this car, or that it's severely damaged, or something like this. And so this is one way to find out the way that, to find out if the fitness of BX is dependent on different partners. Its fitness should be fine when it's with A1, but if it's paired with A 1 through infinity, its fitness is probably not going to be what we would expect. So we could then sort of draw some different scenarios depending on what happens with these intermediates. So the fitness of Bx when it's paired with A1 and A12 could be higher because all the intermediate pieces are there. But then depending on what happens when you start having none of the intermediates, you could get like either a plateau in fitness or worse fitness over time if you pair them all up that way. Now, if there's no coevolution, if these cars have no effect on anybody's fitness, then you would expect to have like a solid line, because then you would say that Bx has the same fitness regardless of who it's paired with. So that's essentially the null model. And there's a couple common models of coevolution that mostly have been tested in antagonistic interactions, and they might look like this if we saw those patterns. And so we did this experiment in my evolution experiment with my evolution experiment. And in this case, what we did, I'm going to show you the methanogen results first. So what this is we took the methanogen from 1,000 generations, and each of these panels is a different co-culture. So we have, this is a co-culture, this is another one, this is another one. So we took this methanogen from 1,000 generations and it paired it with disulfur vibrio from its past and from its future at different time points. And we made all these different pairings and we measured their growth rates. So when we look at this the first time, it's like kind of exciting because it looks like these, the fitness is going up with when the disulfur vibrio has evolved for longer, which could indicate coevolution. But fortunately for science, I think I included a control, which is to account for general evolution. So like, what if these are all increasing just because the disulfur vibrio has gotten faster and faster and is making the methanogen go faster? It may have nothing to do with the methanogen adapting specifically to like the disulfur vibrio at 1,500 generations or something like that. So to test for that, I took the ancestral methanogen and paired it with the exact same disulfur vibrio populations. And that is plotted here. And so you can see that they're very much overlapping with each other, the gray lines and the orange lines. So what this means is that there's not really any evidence that the methanogen has been adapting to specific disulfur vibrio genotypes in these cultures. Instead, it looks like if the disulfur vibrio has evolved to grow faster, it seems to just go for the ride, and the entire culture seems to grow faster. I mean, there are some we could go and analyze each of these separately, like there's some kind of potentially interesting thing here, but really there's not a big difference between the controls and the experimental. Okay, so I've done basically the exact same experiment, this time with the disulfur vibrio. So now I take disulfur vibrio at 1,000 generations and pair it with its past and future partners. And this is what I get when I just look at that data. There's definitely some flat lines in here. So this looks like perhaps no coevolution, probably no coevolution happening here. There are these little blips that I need to look into to make sure that those, I mean, this is the output of a statistical model, but I still, want to know about them. So it seems like that coevolution, if it is occurring, it's that the disulfur vibrio is perhaps adapting to just like its partner at one time point or something, and not in some kind of like way where it's like constantly co-evolving or adapting to its partner. And if we add the control, well, it gets just a little bit more complicated. So you can see that here in the top that it's always causes the co-cultures to grow faster, but it's pretty much the same effect regardless of how much the methanogen it's paired with has evolved. In other cases, you know, it looks like there could be something specific, a specific change like this, where they seem to do better with that particular methanogen. So I think the for sure conclusion that we can draw from this is that it's not, most of the time, it's not co-evolving, but it might be occasionally, the disulfide might be occasionally adapting to some specific aspect of the methanogen it's evolving with. And so if we kind of look at these little experiments that I've presented together, what can we say? Both species adapt and they can cause the cultures to have faster growth. That was kind of shown in one of the first graphs. This adaptation has been accompanied by the loss of a key trait that isn't essential for disulfide to survive by itself. Whether the two species are adapting to each other in the first 1,000 generations is still a little bit unclear, but I would definitely say that they're probably not adapting to unique features of their partner. I think our local adaptation data so far does not show that, especially the part that I showed you just now. We think that sometimes in this last, in this coevolution experiment, it looks like this time shift experiment, it looks like maybe there are particular cases where disulfide adapted to some aspect of an evolved methanogen, but coevolution is not the main mechanism of evolution going on. And it seems that there's a difference between how they're adapting to this mixed environment versus non-mixed environment. Okay, so no, time. All right, so the other thing I was just going to tell you is that we've also been looking at the population genetics in these to see if we've sequenced everything over time, and in both co-cultures and the things that evolved alone, and we're comparing the population genetics of them. And so I wanted, which is a project that is ongoing and relates to this new project I'm starting, which is on eukaryogenesis. And I have 2 postdoc positions available. And what we're testing is this idea that you have a bacteria and an archaea that were interacting and dependent on each other because of the exchange of hydrogen. It's kind of an old hypothesis, but this idea that this had happened for like a long time and they became more and more dependent, and eventually one of them swallowed the other and one of them became the mitochondria. And this is a new area for me. And what we're going to be doing is a lot more looking at our genomic data, making mutants, sequencing much further out along the timeline to see if what kind of adaptations occur that would cause them to become more dependent on each other, and if they continue to become more dependent on each other. And also we'll be looking at symbiosis. So in my experiments, the species do not clump up together really, except when they maybe towards the end of a growth cycle. So we're going to we're using that to our benefit to be able to vary and simulate symbiosis and see how maintaining physical contact, subdividing populations into these groups where they maintain physical contact, how that affects evolution. And to do that, we're going to be making a mathematical model. This is in collaboration with the Kerr lab. And we're going to be sort of replaying some evolution experiments to see if we get different outcomes from what we already have observed by simulating symbiosis versus not. And so there's a lot of work to do, and I'm really excited about this project. It's going to be like a big change for me to think about something that happened way far in the past. And I'm hoping that I can get some people to come and work with me on it. And so if you're interested, please send me an email and let me know. We have been searching for a little while, and anyways, we're hoping that somebody will be interested in the project and let me know, and we can talk about it some more. And so this work has been done by 4 different technicians that are listed here over the years. Blanca just started graduate school in Kansas, and she was the most recent one. And a lot of undergrad, I have a postdoc who's been working on the genomics work, Yimei Shea, and lots of undergrads. Not all of them are listed here that have done this work over the course of several years. So thanks for listening, and I can take some questions if you have them. Thanks. Oh, is that, oh, that's clapping. Okay, I'm not, I have still learning about what you do in Zoom. So yeah, thanks.
That was great. We actually have a few comments and questions in the chat window already.
Okay. How do I find that?
At the bottom of your screen, there should be a button for chat.
Oh, yes. Normally, oh, it's up at the top. It got moved to the top somehow.
Oh, because you're sharing your screen. Okay.
Yeah.
That's right.
Okay. New share, participants. No? Why? Oh, there. It's in orange, so I didn't see it. Okay. Oh. Charles, okay, so what about expression levels of the DSRC protein? Joe was asking. We don't know, actually. I don't know if anyone has done, we haven't done gene expression profiles of the whole cultures, so of all cultures, so I don't know. But that's a good question.
Yeah, I'm just, thinking that that'd be a great thing for you to do, because basically when you do genomes, you just know who's at the party.
Yeah.
But you don't know what they're doing.
Yeah.
Right? So what we found with our iron bacteria was that when we did the gene expression, we got a much fuller picture of which variants were important as compared to just, variants that, went through a selective sweep, but actually don't seem to have anything to do with phenotype.
Yeah, that's good to know. Yeah, at one point I did have like an ambition to do that work, and then I was having so much trying to learn basic stuff to do the genomics itself that the idea of then expanding it to that seemed almost impossible at the time, but it would be nice to get back to that. So, and Joe basically is also mentioning something about, I would have expected that there would have been some differences. Maybe this is because the environments are so similar. Oh no. And now Joe had to go back in time to the March on Washington. Good to know.
The key part of that question was the idea that maybe you might see differences if you gave the pairs a novel environment. Mm-hmm. So if the notion is that they've co-evolved, one way that you could show that is by giving them something new.
That's something I hadn't thought of, and definitely worth thinking
together
about.
For a long time, then that would be good evidence that there was coevolution. So that's just an idea. I mean, I just make stuff up, as you can see.
That's fine. That's fine. And also, like, you had lots of thoughts today, Joe. Maybe you always do. And I'm just not used to seeing them. So this may bear on the old ecological theory concerning plant associations, the notion that there were oak, maple, or beech forests. They lived in the same places because they had the same needs, but there was not much relationship between them.
Yeah, that's the old idea that these communities, were there because they had to work together, like in your case of mutualism. But it turns out that idea just sort of fell apart. It was really the fact that they were there because they all had the same needs. They really had little to no interaction with each other. Yeah.
No, maybe it could be kind of something like that. And I think it's good for us. I mean, of course, at first I was kind of disappointed. I didn't see a lot of coevolution, right? But the more I think about it, the more we need to know and have these examples, right? In order to have a realistic understanding of biology. So Rohan is asking, if the D and M bacteria physically stick together in the treatments. Actually, I accidentally started this experiment, not knowing it, with some kind of variant that didn't have a gene that allows them to stick together. Also, I think I quickly selected against any sticking together properties because they're in this tube and I have to transfer them with a needle and syringe. So if they stick to the wall of the tube, there's no way for me to transfer them. So if there was any kind of overall stickiness, I think I accidentally selected against it. So yeah, so they don't physically stick together, which is a bummer in some ways. A lot of people think that only the ones that stick together are relevant, but I actually kind of think that it allows me to do lots of other kinds of experiments. So, okay. And Dave Queller is asking that at 1,000 generations, the results showed few differences when combined with various generations of other species. Is that related somehow to the issue of uncheatability? It might be. I mean, it might be that there's just this, there's no selection. I don't know. For me, it's so much easier to think about this if one species was like, you know, harming the fitness of the other, and then you would want, you know, it seems like selection would be easier to see where there would be selection to kind of overcome that, right? And this is why I guess I kind of like thinking about positive associations because they confuse me so much. So yeah, it might be that there's yeah, not selection because there's no ability to cheat. Yeah. And then Chris Marks is asking, have you tested fitness of the disulfur Vibrio in hydrogen limited chemostats? There is a lot of work that others did on gene expression in hydrogen limited chemostats. Also, I would say that, you know, when I have the methanogens growing alone and evolving alone, they're not automatically good at syntrophy, actually. I would have thought that would be like the closest case, because in both cases, whether they're in mutualism or growing alone, they're both living with like really limited hydrogen because of the way we have these experiments set up. So I would have thought that adapting to limited hydrogen would just make them automatically good at mutualism, but it doesn't. And so if the basis of exchange is purely Methanococcus wanting the hydrogen to be used and the disulfur Vibrio wanting to use it, then increases in uptake of hydrogen use would speed the whole thing up. We do have some competitive fitness assays I'm still working on. We used your FreakSeq, Chris, we used your FreakSeq method. So we're working on that. I mean, it's done. I'm trying to find the time to analyze it. Yeah, so I think, Chris, tell me if I'm wrong, but I think what you're suggesting is maybe the reason I'm not seeing things is because it's all about hydrogen use potentially. And so maybe there's not that much in terms of the interaction that could be changed. Yes, the BreakSeq. We also use the BreakSeq pipeline, which we like to use. Yeah. Thanks, Joe. Any other questions that I can answer? Okay, Mark Servetnik says, does the genomic analysis give any insight into the increased growth rate over time? There are a lot of different genes that have been mutated. There's like outer membrane porins that are essential to growth. There's sodium, things that involve sodium, like transport and or sodium dependent processes. There's a lot of them, but I think what we really need to do is to start making mutants with some of these, mutate to test whether some of these are beneficial or not. Yeah, there's a lot of things to look at basically and try to put together into some kind of cohesive idea. So there's nothing specific that sticks out. Anything else? Well, thanks for taking the time to listen to my presentation and give me some ideas and good questions to think about over time.
Thanks for a great talk, Kristina.
Yeah.
Yay.
Yay. Thanks. Good to see you all. Yeah.
Good to see you too.
All right.
All right, and this is recorded, and I will be posting it on the page with the schedule.
Great.
All right, so feel free to share it with anybody who wasn't able to make it.
Great. Thanks.
All right. Thanks, everyone. See you next time.
Yeah. Bye.